This is your pre-lab video for the week of March 14th. We'll start with just a couple of announcements. As a reminder, no shorts or open-toed shoes. I suspect this week isn't going to be an issue with the snowpocalypse or whatever is coming this weekend. But please do not come to lab with open-toed shoes or shorts. If you do, we have shoe covers and we also have scrub pants that you can put on, but you won't be allowed to be working with any compounds if you're wearing shorts or open-toed shoes. You're going to continue testing your compounds this week. Please be sure to treat all compounds as if they're toxic. So you should be wearing gloves. Don't touch anything that's touched the compound because the compounds represent possible chemotherapies that could interfere with cell division and cellular processes that are common to your cells, not only cancer cells. On Friday, we're going to have our last workshop. This is the extra credit series that we've been doing. We've covered how to make a CV, what to do with an MCDB major, how to get into medical school, how to get research experience. And this week, we're going to talk about getting into graduate school. And by graduate school, what we mean is a PhD program in the life sciences. So we'll have a variety of different people who are getting or were recently admitted to a PhD program in biochemistry, IFI, eBio, MCDB, and possibly chemistry. But they're there to help you understand the application process and the things that worked for them. This is a great opportunity for people who like research and aren't sure what they want to do. PhDs are not out of reach, but you still need to be almost as competitive as medical school applicants. So we're going to help you with that this Friday at 12:30 p.m. Let's take a look at the schedule for this week. There is no assignment due this Sunday, March 14th, because we have our exam on Wednesday, the 17th. The exam will be held at the same time as last time during our lecture period. I'm going to open it about 10 minutes early so you have a full hour, and I'll leave it open 10 minutes late. In other words, it'll be open from 12:30 until 1:40, and you have any 60-minute period within that time frame to take the exam. The exam study resources page has been populated with a number of different resources to get you practice not only with the basic information that's presented in the lab and in the Play Pause It videos or the perusal readings, but also importantly with critical thinking exercises, which we'll focus on in a review session next week. So I'm thinking what we're going to do is have possibly 2 review sessions, because I want to make sure that we make it available to as many people as possible. I'll send you all a survey this weekend and we'll have it scheduled by Monday morning. So there's no lecture because the exam takes place during the lecture period. And like I said, you're going to be setting up your compound tests again this week. If you're looking for your lab notebook link, it's here. Your last names are all part of the file name. Make sure that you're documenting things day by day. A mistake that people make in Discovery Lab and really in any lab is thinking, oh, I'll just write it down later. And the truth is when you do that, you forget. Writing things down on napkins is a terrible idea, but you'll see lots of people doing it in the lab. Make it practice. Make it a good habit of documenting everything that you're doing during every time that you're in the lab, because you'll be doing that really in any lab that you work in. We're pushing through your research project. So just to review, you submitted a proposal which was reviewed by us and also by your peers in a peer review process. And you should have received all of your peer reviews this past week. If you haven't received your peer reviews, let me know, because that email also contained a lot of information about your particular compound and how you should go about testing it. Next week, your TAs are going to help you create an outline of an introduction section, which will be part of your poster. It's the first part of the poster where you're going to expand on what we did last week with the elevator pitch. You'll introduce the problem, which is cancer and drug resistance. You'll introduce the model organism and your approach to testing and the compound that you're testing. So as a group, you'll create an outline and you can use that outline to prepare a draft. Please consider submitting your draft on the weekend of the 21st. So there's a due date posted for March 21st at 10 p.m. This is an optional assignment. Sunday is the beginning of our academic pause week, and we want to be respectful of that. That said, I think getting feedback on your introduction section is going to be very, very helpful for you as you move forward with your poster. So you don't have to submit it, but we're encouraging you to do so. We'll provide edits and feedback on your drafts, and we'll also grade it. I'm going to put the grade in the grade book, but if you and your group decide that you don't want it to count, just let me know and I'll remove it for every member of the group. I can't break up groups just because of the way the assignments are set up. So you'll just decide as a group whether you want that to count towards your final grade. After that, we're going to move forward and do the methods section, the results section, and the discussion section. You'll be producing a final poster and recording your presentation of that poster, which we will help you with. We're going to teach you how to present a research poster in a format that would be the same as any national conference in science. And then the Cure Symposium, where those will be presented and you'll be reviewing posters from other classes, is going to be held on Wednesday, April 21st at 6 p.m. So this week, as a reminder, the techniques you've already done, but if you feel like you need a refresher, there are videos on collecting embryos, isolating third instar larvae, and setting up food drug vials. The food drug vials look amazing. I was in there today. Today is Friday the 12th. And your larvae are starting to climb up the wall. And in about 10 days, you'll be quantifying survival of those vials and seeing whether your compound worked for the first time. Do remember to put 5 milliliters in the vials. There are a number of vials that look substantially lower than 5 milliliters. So I would use the strategy that Gretchen suggested, which is to measure 5 milliliters using your P1000. Just set it to 1,000 5 times into a single vial with water and mark it. And then you can go ahead and mark it on all of the vials so you know where to stop pouring the food in. Your vials were moved. So today I took the vials out of your boxes because as you accumulate more vials, they're going to start falling down. It's going to be really hard to organize them. And I put them into separate boxes. So when you come to the lab next week, you're going to have the box with your compound, and you're also going to have a box that's divided and has all of your vials inside. So you'll use that box to maintain your vials and to track whether or not they need to be quantified. A couple of tips. So a few of the vials have the flugs, those cotton plugs, pushed way too far in. So you're going to have to remove those flugs to get the flies out. We're not counting the flies, but we have to take the flies out because the flies walk on the walls. And as they walk around, they're going to start knocking your pupae off of the wall, and you won't be able to quantify whether something was there that survived or died. So you need to be able to get those flugs off. Don't push them in super far. They only need to go in a little bit just to keep the flies from getting out. Also, I noticed people are doing a fantastic job labeling all of the vials with the date, your initials, the compound, whether or not it's a positive or negative control. One thing I would suggest is moving your writing up to where the flug is, because you're going to have to look on the wall of the vial to see whether or not there's something inside of the pupae on the wall. If you've written all over the outside of the vial, it's going to be really hard to see behind the marker if there's a pupa on the other side. So try to keep your writing up really high, or maybe over the food where the pupae are not going to be forming on the wall. So just try to move it either up or down. And this is what I'm talking about. Here, these vials are beautifully labeled. I can see that this group marked off where 5 milliliters is. They did a super job getting the food to stop right there. The walls are really clean. I don't see any food on the walls, which can also be an obstruction, but the marker is going to create a problem. So either write way down here and write small, or write way up here. The amount of information here is perfect. They put the date, they put their section number, they put the thing that they tested, and the positive and negative controls are clearly labeled. I did take all of the Sharpies out of the lab. One of the things to keep in mind with Sharpie is that it comes off with ethanol, which is a fantastic party trick. If you're at a party and you want to keep track of your glass, you just bring a Sharpie and you can write on it, and then you wipe it off with rubbing alcohol after. But in a lab, we use a ton of alcohol, right, to wipe down the benches, to clean everything, including your hands. And if you spray it on the vials that have Sharpie on them, the marker is literally going to drip off. And when that happens, it's a disaster because you don't know what's inside of the vials. So this week, you're also going to be setting up a dilution series. So in the first lab, you're going to repeat the experiment you did last time, which is exactly what's shown here. 3 doses of the highest dose of your compound, 1 positive control, and 1 negative control. In the second day in lab, you're going to produce a dilution series. And the dilution series serves a few purposes. The primary purpose is to see if we can get whatever effect your compound is making to go away. And we want it to go away in a controlled manner. In other words, if I decrease the dose of a drug, I want to see the effect go away very slowly, meaning that I can control that drug. If I can't make it go away, where it's either an on-off thing, where it's working so it kills, or it doesn't work, by definition, that's a poison. And we want to stay away from compounds that are like that. So in this experiment in the second class, you're going to be producing a number of different dilutions, and you're going to be testing those different dilutions in an attempt to create a dose response curve. You saw a description of this earlier in the semester when you were creating your proposals, and we'll talk about that in lecture when we return after the exam. So the first thing that you're going to do when you do your dilution series is you're going to take your stock, which is indicated here as tube 1, and you're going to dilute it in a manner called a serial dilution, where you're going to take some of it out and add some solvent. Mix it like crazy. Take some out, add some solvent, mix it like crazy, and so on, to create 3 additional dilutions where the concentration is lower than the initial stock that you were given. You're going to be making 1 to 2 dilutions, which means that the concentration in the next tube will be half as concentrated as the tube before it. This is a screenshot from the worksheet that I made that you'll be getting printed out in lab, and your TAs are going to walk you through how to make a serial dilution. This is a skill that's transferable. You would use this if you're doing Western blots, if you're doing real-time PCR. It's how you create a standard curve to determine the concentration of various things in a lab. So it not only is training you how to create a dose response curve, but also giving you skills that you'll be able to use in other settings in a lab. After you've created those doses, you're going to take those tubes and treat them just as you did your stock. You're going to take whatever volume you were asked to move out of the tube into the food and mix it around. And using this worksheet, you're going to fill in these volumes and the concentrations. And again, your TA is going to walk you through this. There's also a tutorial online on the Canvas schedule that you can check out. Coming up, again, we have exam number 2 on March 17th. It'll be held during the regular lecture period. And your optional assignments due March 21st at 10 p.m. are that introduction draft I was telling you about, and also the drug discovery play pause it or Perusall assignment. I think the people who are using Perusall, I think it's really effective. It's creating a lot of really great conversations and questions about the reading. If you choose to do the Perusall, though, I do encourage you to check the quiz on the play pause it because that'll give you even more practice with the content. Okay, so I'm excited to start seeing results, and I'll see you all in the lab next week.